Good afternoon, class.
Good afternoon.
Today we are going to be introducing a new lesson, which is Introduction to Genetics. And the objectives of this lesson will be as follows. The first one will be to define genetics and some important terminologies as used in genetics. Then we have to explain 4 branches of genetics. And finally, to discuss the history of genetics and Mendelian principles of genetics. Without further ado, let's move on to the definitions. When you hear about the word genetics, what comes to your mind? Who can give us an overview of what genetics is? Anyone?
The study of genetic inheritance.
Exactly. That is very good, Jafet. Genetics is the study of genetics, genetic variation, genes, and heredity in organisms. Then the other term we're going to define is a gene, and define that a gene is a region of DNA, which is known as the locus, located on a chromosome that specifies a trait, and a trait is normally known as a characteristic. Then the other term we'll define is an allele, which refers to alternative forms of a gene that can occur at the same locus or place in the genome. And a good example of this is a person who is a carrier of the gene of tallness and shortness. You find that if the gene of tallness is dominant, that is what is expressed. The next term is locus, which simply means the location on the genome of a gene. And the last terms are haploid and diploid, in which haploid means the condition of having only one set of chromosomes per cell, while diploid means the condition of having 2 sets of chromosomes per cell. So from there we move on to the branches of genetics. The first branch of genetics is known as the classical genetics, which is the branch of genetics that is based on visible results of reproductive acts. And when we talk about visible results, we simply mean those that can be seen by the eye. For example, body mass and height. Then the other branch of genetics is molecular genetics. And you find that this branch encompasses the order and trade of genes and utilizes the methods of molecular biology and genetics. The other form or the other branch of genetics is quantitative genetics, which simply is a branch of population genetics that deals with continuously varying phenotypes. For example, height or mass, as distinct from distinctly identifiable phenotypes and gene products. And this one, an example is the color of the eyes and hair color. So from there, we move on to the next thing. Which is the history of genetics. Under the history of genetics, we will be mainly looking at the most important timelines in the development of genetics. And the first timeline is 1859. And you find that in this year, a scientist known as Charles Darwin proposed a famous theory of evolution in which he came up with the slogan of survival for the fittest, to refer to natural selection. So basically, when he talked about survival for the fittest, he simply meant that those organisms that are best suited are the ones that will survive in a given environment, and the ones that are not suited will be eliminated and therefore will be not able to pass their genetic makeup to the next generation. Then the next timeline is in 1865, whereby a man known as Gregor Mendel experimented on pea plants and discovered the way in which traits are passed from generation to generation. And you find that Gregor Mendel was able to form credible predictions from this experiment, and he also attached statistical evidences on the same. So he was able to test dihybrid and trihybrid crosses in pea plants, and he came up with the results which are used to date. So from there, we are going to look at the 3 laws that Gregor Mendel was able to come up with. And the first law is the law of dominance, whereby some characters tend to be dominant and therefore expressed, while those that are recessive are suppressed. And I gave you an example of height. A person who is a carrier of a recessive gene of shortness, you find that the gene that is dominant, which is the gene for tallness, is the one that is going to be expressed, while the one for shortness will not be expressed. So I'm going to request one of you to read the other 2 laws. Anyone who will volunteer? Anyone?
Law of segregation. It describes how parental factors separate in the gametes cells, that is sex cells. Law of independent assortment. They describe how heredity factors assort independently during the production of gametes.
Very well, Samson. Very well. So you find that the other 2 laws are basically the law of segregation, which entails the separation of genes at the gametes, and the third one is the law of independent assortment, whereby the heredity factors are independently separated during production of gametes. And so from there, I'm going to display for you a chart on Mendel's experiment. And as you can see, the chart has the dominant and recessive traits for flower color, plant height, seed color, seed shape, pod color, pod shape, and flower position. So basically, just as I've said earlier, the dominant trait is the one that is expressed, and therefore it is the one that is actually visible to the eye. You get it? So the recessive trait is suppressed, and usually from these experiments you find that the ratio of expression of the dominant gene to the recessive gene is usually 3:1. So are there any questions, any complaints, or any additions that anyone wishes to make before we summarize the class? Anything? So I take it that you've all understood. Thank you for engaging in the class, and let's meet in the next session. Have a nice evening. Thank you.